Okay, the first major step that we're going to talk about is glycolysis. Again, I said that glycolysis literally means breaking up sugar. We are breaking down glucose in order to create pyruvate. Glucose is a 6-carbon molecule. We're creating 2 3-carbon molecules, literally splitting it in half. This takes place in the cytoplasm of cells, so not the mitochondria. It does not require oxygen, so it is anaerobic. And again, 1 6-carbon molecule into 3 carbon molecules. Okay, so we are right here. Glycolysis, glucose becomes pyruvate. It happens inside the cytoplasm. We're going to get 2 ATP created. So in glycolysis, you basically have 2 different steps. You have an energy investment phase and an energy harvesting phase. So let's talk about what these words mean. Energy investment. If I am investing in something, that means I have to put something in order to get something out of it, right? So we are actually going to have to use 2 ATP in order to get glycolysis going. So when we use ATP, we have to activate glucose by putting into ATP in order to harvest. Now, when we harvest, we are going to create 4 ATP. So overall, we had 4, but we had to use 2 in our energy investment phase for a total of 2 ATP to get created. Okay, so that is our net gain of energy in the step of glycolysis, the first step. So we have substrate level ATP synthesis creating. So here we have pyruvate, right, a 3-carbon molecule. It has a phosphate on it. So our enzyme is going to create ATP by taking a DP plus a phosphate. Remember, a DP is diphosphate, so it has 2. So when you have a DP plus a phosphate, you get a TP. The T stands for tri. So you have 3 phosphates there. And we call this substrate level ATP synthesis. Now, the main thing in all of these steps that you are going to need to know is what goes in, what comes out, and where does it happen. So you're going to see this slide for every single step that we do. And these are very, very, very important slides. Okay, so for your test, I'm not sure when that will be, but for your test, you are going to need to know what goes in, inputs, what comes out, outputs, and where does it happen. Okay, so for glycolysis, we are putting in a glucose molecule. Again, you get this from what you eat. Plants get this from photosynthesis. We are going to need 2 NAD. These are our electron carriers, so we're going to have to put that in. We get 2 ATP because we have to put that in for our energy investment phase. And then we're going to need these in order to create ATP. So these aren't really that important, but you need to know these 3, okay? We have to put in 1 glucose, 2 NAD, which are electron carriers, and 2 ATP in order to get our output. So our output, we just broke apart glucose, so we'll have 2 pyruvate, 2 NADH. We just added electrons. We're called hydrogen electrons. So we just added those electrons. You don't really need to worry about that. And you have 4 ATP output. So again, 4 minus 2 is 2. 4 from output, 2 from input to get a 2 total net gain of ATP. Okay? So first up, the energy investment phase. You do not need to know what all these stuff, you don't need to know what this stands for. You do not need to know about glyceraldehyde 3-phosphate or 3-phosphoglycerate. You don't need to know these. The main thing that you need to know, again, is what goes in, what comes out, where does it happen. And all that the energy investment step that you need to know is that you have to use 2 ATP, okay? So we have our 6 carbon molecule, glucose. We have to add phosphates on it, right? And then we split it, okay? That's what happens in our energy investment phase. For our energy harvesting, again, you don't need to know any of this stuff that happens. You don't need to know. All that you need to know is that you get 2 ATP because, look, we're taking these 2 phosphates, right? We're adding them to an ADP to get ATP. So there's 2 there. Here's 2 other phosphates. We got to do it again. Add it to ADP to get ATP. So that is our 2 net gain. Here's our electron carriers up here, NADH. You add electrons in order to get those. Now, our NADHs are going to go on to our electron transport chain. We do not see these again until the very end. So they got those electrons from glucose breaking apart. They're going to take those electrons all the way over to the electron transport chain. They are not needed anytime soon. Okay, so we have 2 of those. We are going to get these pyruvates, and they are going to move on to the prep reaction. Okay, so we kind of see where everything is needed and where it goes at the end. Now, remember when I said that a lot of ATP in cell respiration is going to be aerobic, meaning that it needs oxygen, but sometimes cells don't have oxygen readily available, so we have to move on to anaerobic, which means no oxygen. And it's also called fermentation. So if you see anaerobic respiration or fermentation, it's technically talking about the same thing, okay? And there's 2 basic forms of fermentation. Lactic acid, which occurs in animals, and then alcohol, which is going to be in plants. So first off, let's talk about lactic acid fermentation. Again, this occurs in animal cells. So we have pyruvate from glycolysis, and it gets changed into lactate. So pyruvate becomes lactate in lactic acid fermentation. That's all you need to know about lactic acid fermentation. It happens in animals. Pyruvate creates lactate. Now, you also are going to get 2 ATP from that. You need to know that as well. Okay, so in lactic acid fermentation, you get lactate, 2 ATP from pyruvate molecules. So fermentation is essential to humans because it can give us a rapid burst of ATP if we need energy in a hurry, but we don't have enough time to complete the whole process of cellular respiration, or we're breathing so hard that we don't have enough oxygen to come in order for our cells to take that. It will then go on to lactic acid fermentation. So in muscles working vigorously means really, really hard, okay, over a short period, fermentation is used to produce ATP. Now, lactate is toxic to cells. Cells do not like it. It changes the pH of your muscle cells, so you get that burn feeling. So whenever you're working out, if you're lifting muscle, if you're lifting weights, if you're running and you feel the burn, that is lactic acid fermentation actually happening in your muscle cells. So you're getting a buildup of lactate when you have that burning feeling. Now, lactate in your cells is going to cause you to be sore later on when you're working out. So it's really important that you do some things to help you get those out of your cells. So drinking water helps the lactate to get released from your cells. Also, things like Stretching and rolling is going to release the lactic acid from your cells. So if you do that, it's not going to build up much in your cells. But do know if you get sore after working out, it's just because you've gone through lactic acid fermentation and have a buildup of lactate in your cells. Now, the second kind of fermentation is alcohol fermentation. Again, this is done by plants and fungus, okay? The main one that we're gonna see is yeast. Yeast is a fungus. It generates ethyl alcohol by fermentation. So this is the alcohol that you drink, okay? So wine, the grapes that are in wine are going to go through alcohol fermentation. Or beer, okay, the hops, the wheat, whatever, the barley is going to go through alcohol fermentation. Yeast does this too when it creates bread. When it causes bread to rise, it does go through alcohol fermentation. So as with lactic acid fermentation, you're going to have pyruvate and you are going to get ethyl alcohol. So pyruvate, you actually get ethyl alcohol and carbon dioxide along with 2 ATP, okay? So this carbon dioxide is what causes bread to rise. Makes it, you know, when yeast is working on the dough and the flour, it causes bubbles. That's the release of carbon dioxide to make that happen. Again, fermentation only creates 2 ATP. There's not many, so it is not that effective, but it is needed in some cases, okay? So again, we have glucose, and we either get lactate or alcohol and carbon dioxide, depending if your animals or plants. What goes in, what comes out, where does it happen? This still happens in the cytoplasm. So glucose goes in, 2 ATP, don't worry about that. And we either get 2 lactate, alcohol, carbon dioxide, 4 ATP for a net gain of 2 ATP. So you might think, well, glycolysis only gave us 2 ATP and fermentation only gives us 2 ATP. How are they so different? Well, fermentation is the end of the line, okay? It does not go on to the electron transport chain. It doesn't go on to the other stages. Glycolysis is going to go on. So this is the very end of the stage for fermentation, whereas through glycolysis with aerobic respiration, you do get to go on and you do get to create those 36 to 38 ATP. It doesn't just stop here for glycolysis like it does